So good morning, or at least morning in California. Welcome to those that are listening live and to those who subsequently will listen to this lecture being recorded. This is the first of a short set of lectures on dynamic energy budget theory, which is being given by myself, Roger Nisbet, from UC Santa Barbara. It's being given with a very useful resources and help from the University of California Center for Environmental Implications of Nanotechnology. So I want to also welcome CEIN people who are listening to these lectures. And the background to this set of lectures, the background to the theory that I'm going to present, derives from the fact that there are many fundamental questions in ecology, ecotoxicology, and of course other areas that relate to understanding the effects of environmental change on populations, communities, and ecosystems. These issues are of societal importance, and we'll talk a little bit about that. And there are so many of them and so many combinations of problems that purely empirical approaches are inadequate. And in the next few minutes, I'm going to argue that theory is required, and the particular flavor of theory that will be covered in these lectures concerns the dynamics of budgets and elemental matter, which are, of course, already widely recognized as unifying themes in ecology. Energy budgets are studied from a wide range of perspectives, and these lectures are going to take very much an ecological starting point. I'm going to emphasize those aspects of dynamic energy budgets that are of ecological importance and of importance for understanding the ecological effects of the contaminants in the environment. And I want to elaborate just a bit on this point that purely empirical approaches are inadequate, because on the face of it, the obvious way to find out what effects particular contaminants have in the environment, or to understand what more generally other ecological stressors have, is to go out and do some experiments. And I've listed in this slide a number of the issues. One is that in ecology generally, there are far too many combinations of organisms and environments to study. Another more pragmatic observation is that experiments and other observations are very much more often cheaper and easier to perform on individuals than on populations. And given that, we need some kind of quantitative framework for relating processes that are operating at the level of the individual organism, in particular physiological and biochemical processes, to processes at these higher levels of organizations, specifically populations, communities, and ecosystems. And finally, in terms of rationale for having some theory, environmental management commonly involves anticipating changes, not just responding to changes, and obviously there are many major changes that it's completely impractical to implement in experiment. The motivation for giving these lectures derived in considerable part from my own research interest in ecotoxicology, and specifically participation in the UC Center for Environmental Implications of Nanomaterials, which is where there is a large group of researchers looking at the effects of engineered nanomaterials entering the environment. And at the bottom of this slide, I've listed a few points that I extracted from a very nice lecture given by Dr. Andre Nell, who is the UC-CEIN director at the initiation of the center. Dr. Nell noted that of a very large number of chemicals, he suggested 50,000 plus chemicals that were registered for commercial use in the US, and this was in 2008. Less than 1,000, I should say, had undergone serious toxicity testing. A full suite of toxicity testing involving animal models typically takes over 2 years to complete and can cost a couple of $1 million. From an ecologist perspective, most toxicity tests involve unrealistic environments. And later in this opening lecture, I'll show some data from the waterflea Daphnia. All the standardized tests involving that animal take place with the animal given perhaps 10 or more times more food than it normally encounters in the environment. So these tests do not directly give you useful information for ecology. And finally, and of relevance to the CEIN participants who are listening to this lecture, the problem is not just 50,000 plus chemicals, it's the fact that when they appear in the form of nanoparticles, that's particles with a dimension less than 100 nanometers, there's a whole suite of new chemical properties relating to shape, size, coating, etc., which means that it's just never possible to cover all these combinations. Nonetheless, it is possible for us to make some progress if we try to identify some general principles that will occur in pretty well all organisms and with many different stressors. And one is the fact that organisms, like everything else, are constrained by the laws of thermodynamics, and we could think of a living organism as living in a thermodynamic non-equilibrium state. It's maintained away from chemical equilibrium by the flow of energy and elemental matter. The term energy budgets or mass budgets, as I will use them in the lecture and as I think most other people use them, relate to changes in the state of an organism associated with various inputs and outputs. An organism changes because various stuff comes in and various stuff leaves it. And when I use the word state, I have in mind attributes like size, mass, life stage, and so on. When I use the word inputs, I'm talking about energy or elemental matter assimilated from the environment, for instance, by an animal, from food, by a microbe through absorbing nutrient across its membrane. There is a huge range there is a huge range of outputs. These include reproductive products like eggs, sperm, babies. They include abiotic products, fecal material, carbon dioxide, nitrogenous compounds, and so on, released to the environment. And these dynamic energy budget models that we're going to talk about essentially describe how inputs from the environment drive changes in the state of the organism and lead to certain outputs. So from an engineering perspective, we're in many ways just thinking of input-output models, albeit complicated ones. And energy or mass budgets appear at different levels of a biological organization. They're relevant at the suborganismal level because the energetic state of an organism can affect gene expression and thereby affect a large number of physiological and biochemical rates. At the level of the individual organism, energy and mass budgets relate performance to growth, development, relate growth, development, and reproduction to the state of the environment, and we'll see some examples later on in this lecture how that's the case. At the population level, energy and mass budgets can result in changes in populations over time, and in CEIN we've seen a number of these examples using microbial populations. More generally, energy budgets can affect qualitative properties of the sort that population ecologists are concerned with. Is the population growing or declining? Is an apparent equilibrium stable? Does the population cycle? What is the age or size structure? What's the relative proportions of individuals of different ages and different sizes? All of these properties can be impacted by energy or mass budgets. More speculatively, there is increasing evidence that energy or mass budgets may impact biodiversity. That's to say the number of different species and the distribution of abundance of these species can be affected by energy and mass budgets. The reason I say that is debatable is there is an interesting debate about what is cause and what is effect. In that case, diversity affects the flow of energy and elemental matter perhaps, and the flow of energy and elemental matter affects diversity. So that is a theme with a large number of open questions. And at the ecosystem level, again, it's perhaps more obvious that the same variables in many cases are used by ecologists studying ecosystem processes as are used by physiologists studying bioenergetic processes. The focus on energy and elemental matter, on flows of energy and elemental matter among groups of organisms is very much what ecosystem people emphasize rather than details related to particular species. And DEB theory, as I've said in the little insert at the bottom of this slide, aims to relate processes at these different levels of organization, but it starts with a representation of individual budgets. So the focus of much of the work that is done in DEB theory is to try and come up with a description of the individual organism, and then to move both up in level of a biological organization to look at the consequences at population, community, and ecosystem level, and inward or downward in levels of organization through to the suborganismal. But we're very much going to be focusing at the level of the whole organism and trying to write down models that characterize the performance of an individual organism. I mentioned a few minutes ago the challenges of one of the rationales that I gave was that this work was relevant to toxicology, and indeed the whole body of dynamic energy budget theory, which, as we'll see in a few minutes, was largely developed by the Dutch theoretical biologist Bas Kooijman, has its roots in toxicology. And that takes me back to my earlier point that there are certain types of experiment that can be done over a very short time at the suborganismal level, and we have at the other extreme experiments that can be done on single long-lived animals or on ecosystems, of which it's only possible to do very few. And a new aim of DEB theory, a new objective that I see becoming increasingly relevant, is to relate these. And again, I've stolen a slide from Dr. Nell at CEIN and adapted it a little bit, but he and his many collaborators at UCLA are involved in a large amount of experimentation involving what is commonly called high throughput screening, where a set of very standard, simple measurements can be done tens of thousands or even hundreds of thousands of times in a day using a robotic system. So we get large bodies of rather limited information on let's say microbes, or perhaps even just parts of just individual cells. And at the other extreme, as I've said, we get the whole organism and ecosystem experiments from which we can get very little data. A recently emerging aim in DEB theory is to ask how can we use this huge body of data that is emerging from these high throughput experiments and use them to get maximum ecological insight. The standard argument, and the argument that underpins a lot of our work in CEIN, is that we use the high throughput information to select the best systems for experimentation at these higher levels. But I think the DEB models open up the possibility of us also being able to obtain some, or attempt to obtain some general insights from these experiments at the very low levels. And I put that slide in here because this is a point I want to return to in the last lecture in this sequence, which will specifically be about toxicological applications of DEB theory. Finally, and in this broad introduction, I note that what distinguishes a living organism from a bag of chemicals, if you like, is the fact that the composition is in some way regulated. That is, homeostasis is the key. Homeostasis is a key property of any living organism. The fact that it regulates the concentrations of its constituents. Formally, I've identified the points here we need to think about. At different points in the course, we'll think about stoichiometry, which is the fact that we don't just need to consider the flow of energy, but of specific elements, carbon, nitrogen, phosphorus, and so on. I've already mentioned the laws of thermodynamics and homeostasis. And of particular concern within DEB theory is the fact that organisms make what I'll call decisions. That is, with certain amounts of energy mobilized, they can use it for supporting growth, supporting reproduction, enhancing the immune system, whatever. They have a range of choices that they make, and we want to model it. And this slide emphasizes components that apply to all taxa. I say here from bacteria to whales, and that's actually true. We have one participant in Transcheck who did his PhD on a DEB model for whales and is currently working for a CEAN on models of bacteria. So there really are these common principles that extend across a broad range of organisms. I'm going to now try and make more concrete some of the problems that we're going to address by looking at some data from one or two particular organisms. And those who know me know that I have misspent much of the last quarter century looking at the ecology of one particular organism, the waterflea Daphnia. And I'm going to start by showing you some data from this beautiful animal. The waterflea, the picture there, is about 2 millimeters in length, and she's carrying a bunch of eggs in a brood pouch on her back. And if you start thinking about this particular animal and say, why should we care about Daphnia other than the fact that I've misspent a lot of time looking at them? I think there are three primary reasons why Daphnia are of interest. The first is they perform an important ecological function. They, in many situations, control the density of phytoplankton in temperate water bodies, and they can be responsible for generating clear water in many lakes. So Daphnia and filter-feeding crustaceans like them are important components of zooplankton. So we've chosen a model animal that is ecologically important and not just physiological convenient. The next reason, which is good for a modeler, is that there's lots of data. Very large numbers of people have studied Daphnia at all the levels of ecological organization that I mentioned. And as I already mentioned, they're also widely used in ecotoxicology. The US EPA, OECD, and various other organizations have standardized tests involving Daphnia, specifically involving mortality and reproduction in Daphnia, that have been used to characterize a large number of tests. And recently, there has been very substantial growth in the data available on standardized tests on using nanomaterials on Daphnia. So for a variety of reasons, it's interesting to use Daphnia as a source of data. So what do we need to know about the biology of this particular animal in order to understand in order to understand its biology? And on this slide, I have listed some of the well-known properties of Daphnia. They're small crustaceans. For much of the year, they reproduce parthenogenetically. I say something about their food. I say something about the fact that their growth involves a series of instars separated by molts. There's a bunch of complicated biology that takes place at the intermolt, and so on. And if I talked to a physiologist, an ecophysiologist, they would say this slide is far too simple. That is a gross simplification of the biology of Daphnia. On the other hand, if I want to be able to move up to the ecosystem level, it's already a lot of information. I really don't want to be modeling all this detail on every animal in a population. So at the organismal level, when we put models together, we're going to have to do a lot, make a serious effort at a model simplification. But the problem is that if I go to the other end and talk to an ecosystem person, they say, well, you can't just think about Daphnia from this perspective, because Daphnia are living in a lake or a pond. There are several major energy and elemental matter flows, and I've just indicated in these slides a really simplistic representation of the flow of carbon and the flow of phosphorus. And they would say that the population dynamics of Daphnia are going to be influenced by the throughput of carbon in the system and the cycling of phosphorus in the system. And again, most limnologists would say that this characterization in particular of the phosphorus cycling is way, way simplistic, that what goes on in a real system is much more complicated than that. So we're walking a fine tightrope when we're trying to define the level of detail to put into our model. On the one hand, we want to put enough information in that we can do something useful. On the other hand, we're clearly going to have to simplify at both the ecosystem end and at the suborganismal end in order to get something tractable. And many of the judgment calls that have to be made along the road relate to deciding what is the appropriate level of organization, or appropriate level of detail, I should say, for the individual organism. In this first pair of lectures, what I'm going to do is take the absolute minimal representation of a single organism. I'm going to focus on just one material. I'm going to say that an organism is really just like a bag of carbon atoms, and see how far it takes us. I'll end the second session this morning by saying something about the limitations of this approach, and that will take us at the beginning of next week's lecture to start considering slightly more elaborate treatments, which will be the starting point of serious discussion of Bas Kooijman's dynamic energy budget theory. So let me now show you some data relating to what individual Daphnia do, because looking at these data gives us some indication of what we're ultimately requiring our model to explain. And I want to focus on several quantities: food quantity, food quality, what happens when contaminants are added, and I will make a very quick mention of nanomaterials. The first, perhaps obvious point is that a food quantity, the amount of food they get, impacts growth and reproduction for Daphnia. Calories matter for Daphnia as for everyone else. The two charts on this the two charts on this figure show in the top right data on Daphnia growth. The x-axis is age, the y-axis is length. Growth at 3 food levels. The lowest is not surprisingly, the animal getting lowest food. The blue curve at the top is the animal getting the highest food. These animals are representative of a very large number of species that exhibit what is called indeterminate growth. That is, if they get more food, they grow bigger. Likewise, if they get more food, they reproduce more. The bottom left picture again has an x-axis, which is age, but the y-axis is eggs per day, and we see there is actually a much larger variation in eggs per day in response to food, with the color coding of these three pictures matching the one on the top right. Food quantity also affects mortality. The data in the figure that I'm showing here comes from a series of survival experiments performed by Ed Macaulay at the University of Calgary, and these are estimates of the mortality rate, that's the risk of mortality per day from cohorts of Daphnia studied in the lab in conditions of low food and high food. The first and most obvious observation is that the animals receiving high food levels experienced higher mortality. That's to say, if they ate more, essentially they lived life at a faster pace and died earlier. And that is a very common observation across a wide range of species. Semi-starved mice live for much longer than mice fed all they want. And this is a theme that I won't cover in the course, but for which there is a well-developed, elaborate, and a rather convincing set of explanations in Bass's DV theory that is discussed in his book, which I'll mention towards the end of the course. Also noteworthy, because it matters for many ecological applications, there is an effect at very low food levels, and in particular for these animals at the lowest food levels, the juveniles are susceptible to higher mortality. And that is the direction perhaps we find most of us find more intuitive, that at low food the animal will be in some sense more fragile. And that turns out to be of considerable ecological importance, this mortality of juveniles at low food, and more speculatively a problem in natural populations. It's not only food quantity, it's food quality that affects the performance of individual Daphnia. And these curves, all that really matters here is that they're different. They're animals fed algae. They're Daphnia again fed algae with different C to P ratios, different C to N ratios, and you see very different performance. The bottom of the take-home message is that having low quality food inhibits growth just as much as having small amounts of good food. So that is something we will have to take account of in our model. And the next slide shows that stressors, specifically contaminants, can impact rates of growth and reproduction. The top right slide is another growth curve for Daphnia with highly contaminated environment here, highly contaminated environment here, low contaminated environment there, and similar situation for a reproduction. I mentioned translating across levels of organization. There are a lot of data on population dynamics of Daphnia. These are natural population data, and one of the things we have to understand is why over a year a Daphnia population, typically through the summer, commonly, typically is not fair, through the summer achieves either a relatively stable equilibrium or has some cycles with relatively small amplitude. In the lab, you get an even bigger range of population dynamics. These are experiments again from the Macaulay lab a little over a dozen years ago, and all that matters here is you can get different patterns of cycles. In the picture, green represents the algal food of the animal, blue represents the Daphnia populations, and you get something like the classic prey-predator cycles. We want to be able to understand these on the basis of dynamic energy budget models. And finally, contaminants affect population dynamics. These are data from a paper published over 20 years ago by Borgman and some collaborators, where cadmium was added to large mesocosms containing Daphnia. And again, for this lecture, the details are not important. The point is that at relatively low levels of the stressor, you can see substantial effects on population dynamics, including one example, the bottom left figure here, where the continuous curve is the Daphnia, and after 2 cycles they go extinct, and the algae recover and continue to do just fine. So there is a range of population level phenomena that we ultimately want to understand, and the hope is that these models can help us. I want to say just a little bit more about energy budgets in natural environments, because there has for a long time been recognition, again in understanding ecological stress, that energy budgets give us at least one clue as to what's going on. And ecotoxicologists, again for a long time, have made use of a concept that they call scope for growth. And what they do is they take an animal that is living in a natural environment, they do some experiments to understand how fast it is assimilating energy from food. These can be performed in the lab. They do measurements of its respiration rate, and the scope for growth is defined as the energy absorbed from food minus the respiration rate. It reflects the energy that is available for growth and reproduction, and in many ways it can be thought of as after-tax pay from the food. If you think of respiration as a tax on the income from the food, scope for growth is a measure of after-tax pay. And for people who have read the literature, or for the people here at UCSB that have been to my previous classes, scope for growth is just the same as what is sometimes called a net production. And it's often correlated with exposure to contaminants. And my favorite example of this comes from data on the marine mussel, a Mytilus edulis, the blue mussel. And the next slide has data on the effect on spatial variability in scope for growth in marine mussels from a range of sites along the eastern part of the United Kingdom. On the right-hand picture here, the x-axis represents scope for growth. 0 means the animal is at the break-even point. 25 or 30 in these units means you have an animal that has a large surplus of intake over a reproduction and over respiration under lab conditions. The y-axis is geographical location sketched out to match these sampling sites on the eastern part of the UK. And there is a huge variation. In the sites on the southeast of England, the animals were scarcely breaking even in terms of scope for growth. From the sites in the far north of Scotland and in the Shetland Islands, which are halfway between Scotland and Norway, there was a very large scope for growth. It turned out that there is a simple intuitive hypothesis about that. If you look at the distribution of population in that country, which is nicely indicated by a picture of the night sky, and I want to thank Eric Muller for giving me this slide, then where red represents densely populated regions, the southeast of England is very densely populated. There is very little population in the north of Scotland. Moreover, the bottom right of that picture which is the coast of France, reflects the fact that the separation of the UK from the continental Europe is very narrow. At that point, it's a couple of 100 miles by the time you're up in this region, and much of this region contains waters that are open to the North Atlantic. Some further evidence that, that's just correlated, some further evidence of potential causality is summarized in the next slide, taken from a couple of other papers that essentially show that from a few sites out of the large number I showed, the body burden of certain contaminants in the mussel, that's the amount of contaminants in the tissue of the mussel, again correlated well with scope for growth. So the basic idea that we'll develop at different points through the course is that sublethal levels of something nasty in the environment can potentially reduce your scope for growth and hence impact organismal performance and hence impact population dynamics. Having sketched through a couple of examples just to give you an indication of the sort of data that we're thinking about, I want to summarize the objectives of the remaining lectures, and they're summarized on this slide. My primary aim is to introduce the principles of dynamic theory in a maximally simple context. That's to say, to use the minimum of formalism to get across the basic concepts that underpin much of the more elaborate theory. Second, I want to demonstrate how DEB theory can be used to relate information on processes at different levels of organization, and in particular I'm going to focus on the link from individuals to populations. And third, my aim is to illustrate how dynamic energy budget theory gives some added value to data in ecotoxicology and to suggest something of the potential in nanotoxicology. And that takes me back to the slide from Andrew Nell that I put earlier. By this added value, I mean getting increased insight from information like that he and his collaborators are getting at UCLA from high throughput screening. In terms of learning outcomes, I would hope that by the end of this short mini course, if you work through this material, you would be able to critically read and evaluate papers that use DEB theory, even if you've not done the detailed work that's necessary to use it. I would hope that you're set up to study in depth the principles of DEB theory if you want to, and in particular I know there are a number of people potentially listening to these lectures who in 3 or 4 weeks' time will start working on the web-based course that Bas Kooijman from Amsterdam is leading. That is a much more in-depth course than this one that requires focus on the detail of the DEB theory. There is a rather low level of rigor in what I'm going to do. There is a high level of rigor in Bas's course, but the trade-off is that I think that the material here requires perhaps 15 to 20 hours of work to get on top of, and Bas I think estimates around 200 hours to get through the material in his class. So this is very much preparatory for that, and I've mentioned that at the bottom. Specifically, what I'm going to do is, in the remainder of this session, in a few minutes we'll have a 10-minute break, but after the break I'm going to introduce what I call dynamic biomass budget models. They're almost a precursor to dynamic energy budget models. They're really simple models based on mass balance, but they're actually remarkably powerful, and I want to demonstrate some of the ideas that way. Because for many purposes, biomass budget models are all we need, and we don't need to get too absorbed in the complexities of DEB. Then, in the 2 weeks that follow, which actually means a total of 4 40-minute sessions, I'm going to work rather carefully through the basic concepts involved in Bas-Kooijman's dynamic energy budget theory, setting out let's say a slightly naive, boneheaded perspective on the underlying concepts, and mentioning a few of the applications. And then the final two 40-minute sessions on the 21st of February are going to be specifically devoted to ecotoxicology and nanotoxicology. So I'm going to talk there specifically about applications to one particular form of environmental stress. So, having said these, it's almost exactly the 40 minutes that I planned. I'm going to now break for 10 minutes for those who are watching live, and at 9:50 we'll resume with a lecture that will give a little more detail on biomass budget models. Thank you. Okay, so welcome back to the second part of this morning's lecture. And in this lecture, I'm going to put a little bit of mathematical flesh on the idea of biomass budgets. I'm going to write down carbon balance equation for a single organism. I'm going to introduce a particular representation of the growth of a single animal called von Bertalanffy growth. It's got a long history in many areas of biology, especially in fisheries, but it's also critical for Kooijman's dynamic energy budget theory, so it's a useful foundation for that. I'm going to talk about the steps we jump through in carefully formulating a dynamic biomass budget model, because they have their analogues in full DEB theory. And then I'm going to talk a bit about the decisions that an animal has to make in terms of, in particular, growth versus reproduction. But again, when in the more full DEB theory, there is a larger range of decisions that need to be made. I've taken much of this material from a book Bill Gurney and I wrote an embarrassingly large number of years ago. I'm going to put a copy of the relevant chapter 4, on the web page for access to people who are listening to this class. I want to say at the start that in putting this lecture together, I had to make a decision, which was that many of the quantities I'm going to talk about have interpretation which is very similar to that is used in DEB theory, but not identical. And I had to make a decision about notation, whether I was going to use similar notation or different notation. I decided that the differences mattered, and for that reason I'm using different notation in this lecture, the same notation actually that we used in the book. And later on I'll point out some of the similarities and differences, but it avoids me having the same symbol with two meanings in different lectures, but the downside is there is notational proliferation. Suppose I want to write down the simplest possible balance equation for an animal, and although the ideas I'm talking about apply to many organisms, I'm going to exclusively focus on an animal this morning. I'm going to assume that its composition remains constant, and again, this is a theme that will recur through DEB theory. It's what Bas-Kooijman calls strong homeostasis, but it's an extreme version of it. I'm going to assume that the composition of the animal stays constant, so I'm only going to focus on one element, and that is carbon. I'm using the symbol W to represent a body mass of carbon, and carbon mass changes for 3 reasons. New carbon enters the animal through assimilation of food. It's excreted or it's respired as CO2. And if I use the symbol W to represent a body carbon, then I can write down a balance equation which simply says the rate of change of the amount of carbon in the animal is the rate at which it ingests carbon from food, less the rate at which it loses carbon through CO2, less the rate at which it egests a carbon. And now I can incorporate an absolute minimum of biology, but again with ideas that will carry over into the more elaborate DEB theory. I'm going to assume that the primary component of egested material is a fecal material, so I'm going to assume that the rate at which the animal egests material is proportional to the rate it feeds, and I use this symbol alpha with a subscript E to be the constant of proportionality. For respiration, I recognize that there are two components that are potentially important. There is a basal maintenance rate. An animal has to expend a certain amount of energy just to stay alive, just to keep organs intact, to maintain ion gradients across membranes and so on, to keep all the minimal physiology functioning. It has to expend energy, and for an animal with aerobic metabolism, this leads to CO2 production. I then recognize that all the processes we're talking about, growth, development, reproduction, and so on, have overhead costs, and there's certainly loads of experimental data to show us that an animal that is growing faster, that is committing more energy to reproduction, actually respires more. So in this model, I assume that the respiration rate has two terms. There is the basal maintenance rate, and there is a term proportional to the ingestion rate. I define a quantity called the assimilation efficiency, which is given there. It's just 1 minus the two constants of proportionality. And then the balance equation for carbon mass takes the form illustrated at the bottom here. The rate of change of the carbon mass is this pile of constants times the ingestion rate minus the basal maintenance rate, or in simpler form, it's an assimilation efficiency minus the maintenance rate. For many decades, closer to a century, this type of balance equation has been recognized and used as the basis for obtaining a simple model to describe the growth of an animal. Suppose I define a quantity L to represent some measure of the organism's length, a head to tail in a fish, for instance. Then, provided the organism does not change shape during its lifetime, then we can assume that the weight of the animal or the carbon mass of the animal is proportional to the cube of length. And many authors, including Koyman in his DEB theory, call an animal for which that property holds an isomorph. There are then many arguments that say that feeding rate should be proportional to some measure of surface area. That is to say that certainly assimilation of food involves food crossing some surface, and if the animal stays reasonably constant in shape, you could assume that feeding rate is proportional to surface area, so you get the ingestion rate proportional to some measure of length squared. And the basal maintenance rate, it would seem not unreasonable to assume is relatively invariant. If you remember, this is a baby model. We say that a cell is a cell is a cell. It costs a certain amount to keep the cell functioning and to hold the animal together. So for now, at least, we'll assume basal maintenance is proportional to weight, which means length cubed. You substitute these quantities into the equation in the previous slide, and you come up with this equation, a very simple first-order differential equation that describes the growth of the animal, which says that the rate of change of length is proportional to the difference between the length and some maximum size with a constant of proportionality with the units of time. And if you now plot length on this axis, apologies I forgot to label these, and time or age in this axis, that equation tells you that the pattern of growth in an animal looks something like this. It's a curve that rises steeply and then flattens off, and this pattern is observed with many, many organisms. The equation has a long history, much of it preceding a von Bertalanffy, and I've put on this slide a URL to a section of a very useful file put together by a Basque-Hyman called Comments, where he adds additional information as it becomes available relating to the material in his book, and there is a rather nice discussion of the history of von Bertalanffy-like models at the point that I've indicated there. The von Bertalanffy equation gives good fits to growth data for many species. A large number are listed in Kuyman's book, and I think since the book a number of other data sets have been analyzed, and like I already said, it has a long history of application. But there is nothing fundamental about this equation. I wrote down a simplistic version of the assumptions, but they may not be strictly true for many animals. And in fact, there is a rather nice short paper in Nature published almost exactly 10 years ago by Banavar and a group of co-authors that showed, in terms of data, if instead of having an input term proportional to length squared, which meant weight to the power 2/3, you assumed weight to the power 3/4, that comes out of another whole collection of theories. There are many theories that argue that assimilation rate should be proportional to the 3/4 power. You get just as good a fit. And in fact, more generally, mathematically, if you write down a differential equation of the form I've written there, for a range of values of alpha, it's 2/3 in the von Bertalanffy, it's 3/4 in these other models, but for a range of values, you get a reasonable fit to data. And I've copy-pasted here the key figure from that short note to Nature. These are four curves, four sets of data that were fitted by both the von Bertalanffy model and the model with the 3/4 power. The details don't matter. What matters is that the two curves, which I can hardly make out the separation of in these slides, in this slide, do a pretty much equal job and a good job of fitting the data. And in fact, that paper goes on to show a way in which you can construct a data collapse that causes all models of this sort to essentially fit on one curve with a slight rescaling of time. That is a scope, a point that goes beyond the scope of this lecture. What I think that tells us is two things. One is that there is clearly considerable empirical support for something like the von Bertalanffy representation of growth, but that we need a more systematic way of writing down a model. And I'm now going to detail briefly the assumptions that you would make if you just focused on setting up a biomass-based model, and then next week we'll move to talking much more carefully about how that can be done in an even more systematic way using some dev concepts. My starting point remains this balance equation. That lets me make perhaps another general point that when you're writing down virtually any dynamic model of energy or mass budgets, it's, I think, often very useful to just write down the balance equation first before you make any assumptions, because the one thing you know is that the balance equation is true. The assumptions may or may not be wrong, the detailed assumptions, but if you start from the balance equation, you always have a rigorous foundation. I'm now going to make a more general set of assumptions. I'm no longer going to assume that mass is proportional to length cubed. I'm going to say that it's proportional to some power of length to get a general family of models. I'm going to retain the assumption that maintenance is proportional to weight. I'm going to make the assumption that the assimilation efficiency is always constant, and that is one that in some cases we might want to return to, because sometimes the state of the environment affects the assimilation efficiency. But it certainly assumes that I'm only considering one type of food, because self-evidently the digestibility of different food types is different, and so that is an assumption that you might in some situations have to revisit. I then have an assumption about feeding rate that has a property that is common to the model I'm going to talk about and to much of Dev, but I actually think that it's one that is one of the flakier assumptions in much of what we do, and in some ecological contexts it needs revisited. And that is that I'm going to assume that the dependence of feeding rate on size depends multiplicatively on two quantities. The dependence on size and food density is multiplicative. And what I mean by that is that an animal of a given size can feed at a certain maximum rate that is then modulated by the amount of food that's available. But the factor by which it's modulated is the same for small animals and large animals. For those of you who have familiarity with with standard ecological jargon, we can go further and assume that if F is a measure of food density, that there is a type 2 functional response, sometimes called the Halling type 2 functional response, or the Disk equation, of the form F over F plus a half saturation constant. This equation with slightly different notation is also widely used by Kuyman. The expression of the problematic nature of this assumption that I'm making is that everything I'm going to do now assumes that the half saturation constant is the same for all individuals, whether they're small or large, and it's a subject that we could fill half a lecture with. Obviously I don't want to, and but I'm mentioning it now because when you come to apply some of the DEV theory, you do discover that there are often problems associated with this constant, and it may be that this assumption is the source of part of the difficulty. If we accept these assumptions, the question we then have to ask ourselves is, how does this maximum feeding rate vary with size? And I think, broadly speaking, in the literature, you will find there are two types of assumption. One is the assumption that was already made with the von Bertalanffy growth, that it was proportional to some rate, it was proportional to some measure of surface area. You can then take the assumptions that I wrote down in that previous slide, do a bit of algebra, and that is in the book chapter that I will post on the website. And you derive, not surprisingly, since the assumptions are similar, the von Bertalanffy growth rate. But the important point is you now get a connection between the parameters in the von Bertalanffy growth rate and underlying physiological processes. According to this representation, the constant I previously called gamma, which was the maintenance rate per unit of carbon in the system, the von Bertalanffy growth rate is a third of that quantity mu. The maximum length is a complicated combination of quantities. There is the assimilation efficiency. There is this thing which is the Greek letter xi, which has come out rather small here, which is a constant in the feeding function. There is the functional response, the measure of food availability. There is the maintenance rate, and there is this Greek letter chi that represented a constant in the weight-length relationship. The payoff for writing things out carefully in this way is that if we now want to consider the effects of environmental stress, we can now say which parameters in the von Bertalanffy model would be affected. So we discover that because of some change in environment, there is going to be less food available. That will manifest itself through a change in phi, the functional response. We will see in the fourth week of the final week of these lectures that in many cases the presence of contaminants in the environment affects the feeding rate. That would affect this quantity xi, again reducing the maximum length. We will see that there are many experimental situations that suggest that a maintenance rate, or certainly respiration rate, increases in the presence of contaminants. That will affect this quantity mu. And a change in mu will affect the maximum length. Again, if it's increased, say, through more detoxification costs, Lmax will go down because mu goes up. But so also will the rate of approach to maximum size, because now there is an effect in two places. And if I now go back a couple of slides to the equation for the von Bertalanffy growth, where did it has? Oh, here it is. It has these two constants, but it has no, written that way, there is no obvious interpretation of these and no way of characterizing, of relating them to underlying physiology. And in fact, the fisheries literature is full of papers that describe how gamma and what they call L infinity vary with temperature, acidity, you name the stressor of interest, and somebody has documented how the growth constants for some particular organism vary. If I move back to where I was before, to this simplistic model gives us the potential for some mechanistic interpretation of what's going on, and some way of getting a unified description of the effects of different types of stress. A second assumption that is often made is simply that feeding rate is proportional to biomass. And this is more commonly made among ecologists who are working with populations because they don't want to consider the complexities of changes in size structure, but it's still quite often made. Again, a few steps of algebra, and all the other assumptions in the previous slide, of course, hold. If you make that assumption, you conclude that the weight and hence the length of the animal will simply grow exponentially with a growth constant there. And you might think that is a pretty silly model, but we'll see in a minute that it's silly the way I've written it down, but it is savable, and okay, so welcome back after the break. We've no idea what has happened, but we all lost our Blackboard connection, and it seems from looking at the chat box that many of you are back connected. So I hope you were with me to the point where I finished up talking through this slide. I talked in detail about surface area feeding. I mentioned volume feeding. I talked about the payoff from the systematic formulation being that we could relate the parameters in a phenomenological model to underlying physiological rates. I mentioned this challenge of thinking about how a maintenance rate affected both parameters, and made this comment that a volume feeding was not quite as silly as it might appear. The two models in the previous slide make contrasting predictions. The surface area feeding model makes exactly the predictions we would expect. At low food, you get a von Bertalanffy-like curve to a low maximum size. At high food, you go to a higher maximum size. The volume feeding model, the way I've written it down, implies exponential growth, which of course we've never observed with any organism anywhere, I don't think. But the resolution of that is to recognize that as an animal gets bigger, it allocates energy to growth and energy to reproduction. And to take account of that, what I've done, give me 1 minute, I have lost the pointer. I think I've got it. Yes. To take account of that, we readjust our balance equation, because now the animal's mass changes for three reasons. One is it assimilates energy from food. The second is maintenance. But the third is that it loses carbon in a reproductive material, specifically in eggs. And now there are a range of possible assumptions that we could make, which I don't want to describe in detail. But one is what is called production allocation here, and that is that the difference between the income and expenditure, that's the entire right-hand side of this, the first two terms on the right-hand side of this equation are allocated to reproduction. Some fraction of that goes to reproduction and the rest to growth. And the second possibility is that simply some fraction of ingestion or fraction of the assimilated food goes to reproduction. Sorry, let me say that again. The first example, the first case is that a fraction of the net production, that's the first two terms together, goes to reproduction. The second case is that a fraction simply of the first term, that's the assimilation, goes to reproduction. And you get different equations, obviously, with these two assumptions. However, the story is now rather complicated, because I've said we have two possible forms for size-dependent feeding, two strategies for allocation of energy to reproduction, and I could easily make more. And we need we clearly need an approach that avoids just endless proliferation of options. And to do that, we focus on the qualitative properties of the models, of the four models, before we do any computations. And again, the details are in the book chapter that I will post. But I've summarized on the next slide the primary properties of a very simple model that you can write down with net production allocation. There's a few steps of algebra written down here, which I don't need to talk about. The important point is you finish up with von Bertalanffy-like growth. The expression for the maximum size is exactly the same as the one I had before. And that means that this model predicts that when growth stops, reproduction stops. And that means that as a fish, for instance, reaches its maximum size, it stops reproducing. That is obviously inconsistent with observations on a huge amount of data, which suggests a potential problem with that model before we do any computations. However, there is a way out of that. I assumed in that previous model that a fixed fraction of net production went to reproduction. Suppose the fraction to reproduction depends on age or size. So a fish above a certain age essentially stops growing or slows down growth and puts more to reproduction, or a fish above a certain size slows down growth further than von Bertalanffy would say and allocates to reproduction. And the end result is that you get growth to a maximum size that is lower than the von Bertalanffy size would be. If you continued just the von Bertalanffy, that curve at low food would go up to somewhere about here, and the curve at high food would go up to somewhere about here. So you get a reduction in maximum size and sustained reproduction, and that again is consistent with many observations. And if we go back to my favorite animal, Daphnia, if you look now at data on the allocation of carbon from food to reproduction as calculated from a whole series of experiments, you see data that I've aggregated on this picture here. Actually, the y-axis is proportional to growth, so 1 minus the proportion to reproduction. And what you see is that there's a lot of scatter in the data, but you could fit a simple function through that, and you then take the production model and you run it through growth data, and everything works just fine. You can do a similar exercise with an allocation model. So we're still stuck with this problem that we have a range of options, and as we add elaboration, things will get yet more complicated. And I should have mentioned this slide. That slide just takes you through the allocation it takes you through the fit of the growth data. That takes you through the math with the assimilation allocation, and again things work out pretty well, just fine. And where I want to go with this is that from the analysis that we've done, and I've rushed a little bit at the end because of the delay because of the break in the middle, we have a number of components in our modeling. We have a carbon balance equation, we have a weight-length relationship, we have a maintenance weight relationship, we have assumptions about maximum feeding rate, we have assumptions about the functional response, in particular the argument that the functional response has a half saturation constant that is independent of the environment. We have assumptions about assimilation efficiency and assumptions about the measurement of allocation to reproduction. And I think I've scarcely scratched the surface of it. Things could get more complicated again. And I want to end the lecture with really a point to think about, which provides the segue into the next lecture. The complexity of this lecture indicates that if we purely take what I will call a modeling approach, that's to say we start from data, look at patterns in data, and try and infer a model that we will use in a particular range of circumstances, we're faced with a very wide range of choices, and the more detailed the application, the larger the range of choices. And this raises the question about whether for more practical applications where we will typically have way less data than we have for a small number of model organisms, there isn't a case for trying to develop some general theory. And by theory I mean here some models that are based on what I'll call general design principles that can be applied across a broad range of organisms. And of course, by posing this question, I'm implying that I think the answer is yes. and that a first cut at such a theory comes from Bas Kuyman's dynamic energy budget theory, which will be the subject matter of the next pair of lectures a week from now. I apologize to those that were listening remotely for the break in connectivity, and we'll see how that works out with the recording. And thank you very much for your interest. Till next week. Bye.